All right, I didn't hear anyone join, but there are people here. Can somebody confirm you can hear me?
I can hear you.
Great. Okay, let me see. I think we're on chapter 8. I was trying to load that. All right, I got what we were on conjugation. Let me share my screen here. All right. Are there any questions before we begin? If not, let me show you the syllabus. We are supposed to be on chapter 14. We're not there. We're behind schedule. And it's kind of bad because you're going to have a quiz on chapter 8. And I have to finish up chapter 8 today. There will be no lecture today. I will log in for the first 15 minutes to answer questions, but after that, I will log out. Let me inform you that your unknown notes pages are going to close on Friday, March 8th, and the unknown report is due Saturday, March 9th. In your unknown notes pages, you are graded on that. It will count as a regular lab. So make sure you finished your unknown notes pages. A lot of you have not put up your results to the last test I sent you. So you want to make sure your unknown notes pages are finished by Friday so you can get the 5 points total out of it. Any questions about any of that?
Are we going to have the lab 9 graded in time for the quiz tomorrow?
I'm working on it, and I just noticed that the lab 8, which I was grading before starting the class, is not on the next quiz. In fact, it's not on the quizzes at all. And I do realize why that's the case because we added lab 8 later than all the other labs when we designed the online labs for COVID-19. And so is no lab 8 on any of the quizzes. That's just the way I initially set it up, and I never went in and changed the quizzes. So I'll stop grading lab 8 and try and get lab 9 graded. I will try. There's no guarantee, but I will try. Any questions about any of that? All right. Let's finish chapter 8. So I talked about conjugation where one cell is an F plus cell and it has a F sexual pilus, which actually skewers an F minus cell and then can bring the two cells closer together. The F plasmid and the F plus cell, which codes for the sex pilus, can then replicate itself and send it through the sex pilus into the F minus cell. That will change the phenotype of the F minus cell to an F plus cell because it has the F plasmid, and that F plasmid will code for the construction of a sex pilus in the F minus cell. So we change the phenotype of that cell from F minus to F plus. Any question about any of that? The third way that horizontal gene transfer can happen in cells, particularly bacteria cells, is by transduction. Transduction uses a virus to move DNA around from one bacteria cell to another bacteria cell. And what happens in some viruses is that they will infect the cell shown here and then inject their DNA into the cell. Now, the viral DNA wants to call the shots in the cell. So one of the first things it does is it will encode the destruction of the chromosome. And the reason is you destroy the chromosome, then your DNA is calling all the shots in the cell. And the shots that the viral DNA will call will be to replicate the virus. And so the chromosome bacteria DNA gets chewed up into pieces. And then the viral DNA, we're just going into a little bit of viruses. I haven't talked about viruses yet, will replicate itself. And then the DNA will be transcribed and then translated into viral protein. And most of that viral protein will be encoding the capsid or the protein of the virus. We then insert the viral DNA into the virus. And then the last thing the virus does is it encodes the destruction of the host cell, and that will release the virus. But viruses make lots of offspring, and if they make a mistake, it's not really a problem for the virus, and occasionally mistakes get made in viral replication. And here is one right here, and that is, let me blow this up a little bit. The viral DNA is this red purple, and that right there, this virus got a piece of the host cell's chromosome inserted into it, not the viral DNA. And what happens when this cell virus goes and attaches to a cell, it will replicate, it will insert its DNA genome into the host cell, but that's not viral DNA, that's DNA of this host chromosome. And then this DNA can recombine with this host chromosome, and then this DNA can be expressed by this recipient cell. So this cell has picked up new DNA, which is DNA from this cell. And that is one way that genes can move around in a population of bacteria cells. That's really important if this gene is carrying antibiotic resistance that it got from this cell. Now this cell will become antibiotic resistant. Any question about any of that? All right, let me end this and then let's talk a little bit about the frequency of how often these events occur. Let me blow this up. So if ever there's no genetic variation around, and then the population gets that new genetic variation, the genetic variation has to come from, come on, that has to come from mutation, usually spontaneous mutation. I'm just going to abbreviate that. And this will happen at one cell in 1 times 10 to the 6. Let me go there. We're talking about actually not genes. So for every gene, out of 1,000,000 genes, one of them will be mutated. And if you have a population of 1,000,000, you're going to have one cell with a mutation in that gene. Any question about that? Because we have 1,000,000 cells, and if the mutation rate is 1 in 1,000,000, then you have your mutation. Let me superscript that. I'm having trouble getting that to show. There we go. Now, transformation, oh, why did that get small like that? Oh, that's why. Transformation can spread around genes about 1, and I'm just going to copy that. Didn't mean to do that, but good enough. One, into the fifth cells, or 10 times more frequently than spontaneous mutation. And the same with conjugation. And then transduction. Meaning that if a cell is likely to pick up a new gene, it's 10 times more to happen by transformation, conjugation, and transduction than it is by spontaneous mutation. Any question about any of that? So if a cell picks up a new gene that is pre-existing in the population, it's much more likely to pick that gene up from transformation, conjugation, or transduction than it would be from spontaneous mutation. Any question about that? Now, of course, we're talking about bacteria cell. Transformation doesn't really happen in human cells. Conjugation doesn't happen either, although we have sexual reproduction, which is somewhat similar, but obviously it's very different than sexual conjugation. And then in transduction, in our germline cells, transduction does not happen, at least very often, where a virus picks up a gene and then inserts it into a germline cell. However, in our nose cells, and maybe the cells of our throat as well, transduction probably happens in human cells all the time. But who cares? Because if one cell in the nose picks up a new gene, that gene will never be passed on to the offspring. So for at least evolutionary purposes, the transduction happening in our nose cells and our throats doesn't matter. Any question about any of that? All right. But in bacteria cells, the frequency of transformation, conjugation, and transduction are extremely important. Go back to the lesson. All right, let's talk a little bit about plasmids. Plasmids are small circular pieces of DNA that we call extrachromosomal because they're not part of the chromosome. They do contain genes, and in a sense, they're the same as the bacterial chromosome, that it is circular DNA. Obviously, it's chromosomal, and it's not small. The chromosome can have tens to hundreds of thousands of genes on it, and the plasmids only have a few genes on them. So this one has maybe 1, 2, 3, maybe 4, 5, 6, 7. If these are genes, I don't know if those are. Something like 7 genes on this plasmid. Not very many when you consider a chromosome has tens to hundreds of thousands of genes. Plasmids do carry genes and DNA, and they replicate independently of the cell's chromosome. We've actually shown you that when we were talking about right here, the F plasmid. That plasmid replicated independently of the chromosome, making a copy of itself and then sending one copy into the F- cell because the plasmid replicated independently. The importance of plasmids is that a plasmid is much more likely to be transferred from one cell, excuse me, into another cell, meaning plasmids, especially by sexual conjugation, are much more likely to move from one cell to another. But plasmids are also a little more likely to move in transformation than chromosomal DNA, and a little more likely to move in transduction as well. So plasmids are important because if you have a plasmid, they are more likely to have the DNA move from one bacteria cell into another bacteria cell. There are different categories of plasmids. You don't need to know the names or what the categories are. Just know that there are different categories of plasmids. Any question about plasmids? If not, let's talk about transposons. Transposons are a region of DNA that moves about from one area of the chromosome to another area of the chromosome. Now, in eukaryote cells, the transposon can move from one chromosome to another chromosome. So a segment of DNA that can move from one region of DNA to another is a transposon. Transposons always include the transposase gene, and they also have an inverted repeat on both ends of the transposase gene. It's thought that it's believed, we should say, that the inverted repeat helps the transposase move into a new region of DNA. So the transposase gene literally codes for an enzyme that cuts the transposon out of the DNA. So it cuts it here and here, and then finds a new region to move into, and then cuts open that DNA. And then the inverted repeats help the transposon move into the new location. It's called an inverted repeat because if you look at that, it is inverted from this segment here. There it is, ACT, and here we go backwards, ACT. Any question about transposons? All right, we can have complex transposons, which have DNA in the transposon, such as transposon number 5, which carries the kanamycin resistance gene. When we have a complex transposon, we have the DNA of interest being carried by the transposon, which in this case is kanamycin resistance. And then usually we have a transposon, a complete one over here, and a second complete transposon over here. So there's transposase gene there, and then these are the inverted repeats. Any question about transposons? Okay, transposons are important because they can move around DNA from one chromosome part of the DNA to another part of the DNA, including moving a gene from the chromosome, like transposon number 5 here, moving into a plasmid. And now this plasmid will be more likely to move from this cell to another cell, especially by sexual conjugation, but also by transformation and transduction. And so transposons are important because they can move, well, DNA around from one region of the cell to another. And then that can increase the occurrence of kanamycin resistance gene in the population, because if it moves into a plasmid, then this plasmid DNA is more likely to spread to other cells in the population. Any question about transposons? They're actually first discovered, I think, in corn, and somebody was trying to find out, map location of the gene that was causing speckling in the color of multicolored corn. And they noticed that the gene location was moving around, and they discovered it was the transposon. So transposons do happen in eukaryote cells. I don't know if it happens in human cells, but it probably does, because it definitely happens in corn. All right, let's talk about genes and evolution. Genetic variation, if it's new genetic variation arising in the population, it has to come about by mutation. And I did once mention that 10,000 years ago in the human population, there was only the brown-eyed allele in humans. And then in Northern Europe, there was a mutation, and the blue-eyed allele came into being. And now in Northern Europe, that allele is actually the predominant allele in Northern Europe. But once the genetic variation exists in the population, it can spread around by transformation, conjugation, and transduction. And we're mostly talking about a bacterial population, which obviously has conjugation. And they're more likely to have transformation. Human cells are not good at transformation. That can happen, but it's not good. And then transduction, like I said, it does happen in the nose and the throat cells, but it doesn't happen generally to our germline cells. So it's unimportant in evolution of humans. But in bacteria, it's very important because it happens 10 times more frequently than mutation. And then crossing over is important in genes and evolution because crossing over can not create new genetic variation, but have genes come together that previously were not together, so that new existing pairs of genes can come about by crossing over. And then transposition just kind of helps mix up the gene and then also helps the transformation, especially conjugation and transduction, occur in bacteria cells. And all of this genetic variation drives evolution of the population, and the evolution happens by natural selection. Any question about any of that? So natural selection will act on the genetic variation, giving rise to evolution in the population. All right, that's it for genes and evolution. Any questions on chapter 8? If not, let's move on to our next chapter, which is chapter 14. The principles of disease and epidemiology. As is normal, I give various terms. I've only got 3 here. However, the terms or the objectives of this class, if you learn the terms of this chapter, you'll learn 50% of the material of this chapter. This chapter is really important on learning the terms. So learn the terms of this chapter. 2, understand the relationship between the normal microbiota and its host. 3, understand the stages of disease development. And 4, understand what nosocomial infections are and how to control them. Any question about what we're going to cover? All right. The first terms you need to know is pathology is the study of disease. Etiology is the study of the cause of a disease. For example, the etiological agent of anthrax is Bacillus anthracis. Pathogenesis is the development of a disease. Infection is the colonization of the body by a pathogen or the presence of a microbe where it is not normally found, where you can say it's not supposed to be found. Like E. coli is normally found in your gut and it doesn't cause any problems when it is in your gut. However, if it is in your ear, it can cause an ear infection. And if it's in your lungs, it can cause a lung infection that we call pneumonia. And then, of course, it can also cause a UTI if it's in your urethra. So that's what an infection is. Disease is an abnormal state in which the body is not functioning normally. When we're talking about disease and pathology, you should come to understand that few microorganisms are pathogenic, that few microorganisms can cause disease. Most microbes are not pathogenic, and some microbes are even beneficial to the host, particularly the microbes or the normal microbiota of the host. Any question about any of that? All right, let's talk about the normal microbiota and the host. The normal microbiota is a large community of microbes found in association with all animals and plants. When we're talking about the human normal microbiota, you should actually use the term human normal microbiota because different animals and plants, obviously, have different normal microbiota. Now, there's a lot of overlap in the normal microbiota, for example, between humans and cats, like you'll find Staphylococcus epidermidis on both species as part of the normal microbiota of their skin. However, you can find certain microbes on cats that you do not find on humans. One of them is Bartonella henselae, which is a part of the normal microbiota of cats, but is not part of the human normal microbiota. In fact, if humans have Bartonella henselae, it's likely to cause disease, and we call that cat scratch fever. Any question about any of that? All right. This disruption of the normal microbiota host balance appears to be associated with disease and impaired immune function. For example, you can disrupt your normal microbiota and host balance by taking antibiotics, particularly if you take the antibiotics orally, because not only will the antibiotics hit the pathogen that they're prescribing the antibiotics for, but it will hit your normal microbiota, all of the microbes in your gastrointestinal tract. And that frequently will give the patient diarrhea or constipation, which is a symptom that the normal microbiota host balance is no longer in balance, it's out of balance. Fortunately, it usually reestablishes a balance, and then the diarrhea or the constipation go away after a day or so. Any question about any of that? All right. I don't know if you've ever taken antibiotics and then had diarrhea or constipation, but I had, and usually it goes away within a day. The human microbiota is found in this picture here on the skin. All of the mucous membranes open to the outside, like the eyes, the mouth, the nose, the throat, the gastrointestinal tract, the lower reproductive and urinary tract, especially the urethra. The normal microbiota are microbes that colonize usually the baby, and then once they establish themselves, they usually live with their host for their host, close to their host lifetime. Like when Staphylococcus epidermidis first colonizes the baby, which usually that happens during birth or right after birth. And then it usually will stay, Staphylococcus epidermidis usually will stay with the host for the life of the host. So usually the normal microbiota colonizes the host for about the lifetime of the host. It might be shortly less, but about the lifetime. That's different from a transient microbe microbiota, which may colonize the host for days, weeks, or months, but unlike the normal microbiota, does not colonize the host for their essentially almost lifetime. I once had an example of a transient microbiota, and that was when I was testing myself in the microbiology lab. And so I was doing it every term, just testing my skin to see if I had Staphylococcus aureus. And I never did until one time when I went to visit my aunt, and she insisted on hugging me, and she had a Staphylococcus aureus skin infection. And she probably thought she would be fine because she was taking antibiotics to treat the skin infection. But when she hugged me, shortly thereafter, I happened to be testing my skin, and I had Staphylococcus aureus appear on my skin for the first time. And if you don't know, Staphylococcus aureus is a human pathogen on the skin. And I wasn't too happy about that and said, geez, I must have picked this up from my aunt when she hugged me. Fortunately, the next term I checked my skin, checking for Staphylococcus aureus, I didn't have any. And then all future terms, when I checked my skin, I never had any further episode of Staphylococcus aureus growing on the mannitol salt agar plates. So that was an example where I had a transient microbiota, which for, I don't know, a few days, weeks, maybe even a month or two, I had Staphylococcus aureus growing on my skin. Fortunately, my normal microbiota threw off the Staphylococcus aureus, and then Staphylococcus aureus disappeared from my skin. So that was a transient microbiota. And I never got a skin infection, which I'm pleased to say. It doesn't always cause a skin infection, but if you're a person who frequently gets boils on your skin, it's probably because you're one of the 20% of Americans who has Staphylococcus aureus as part of their normal microbiota on their skin. I, fortunately, am not 20% of Americans. I had it as a transient microbiota. Any question about any of that? All right. The normal microbiota is about 4 times 10 to the 13th bacteria cells on a normal human, meaning a human. And humans only have 3 times 10 to the 13th human body cells. So we have more normal microbiota cells than we do human body cells. And that begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much, because if you go by bacterial numbers, we are. It's just the way the normal microbiota are. This is showing you some pictures. The normal microbiota bacteria, the little round orange ones in the nasal epithelium. Those are part of the normal host, the nasal epithelium. What do you call them? Villi in the nose. There are some normal bacteria in the stomach. There's the stomach lining with the villi of the stomach. And there's some normal microbiota in the intestine. And you can see many types. That looks like a rod. There's another rod. That's a curved rod. I'm not sure what that is. That one, I'm not seeing it now. Someplace in here, there's a cockeye. Maybe it's that one there. And then those are cocci bacillus. Cocci bacillus. That's food in the intestines. We found some unusual or surprising places to find the human normal microbiota. For example, in female bladders, females have a normal microbiota with bacteria living in their bladder. It was thought that the urine in the bladder was sterile until about 2014, but it is not. That females have normal microbiota in the urine in their bladder. And I've looked to see if people can answer the question, do males have a normal microbiota in their bladder? And none of the studies have ever looked at males. And so I don't know the answer to that. There are some differences, and that is males have a longer urethra, so it's possible males may not have bacteria as part of their normal microbiota inside their bladder, but females do. And that's fairly recent discovery, 2014. Almost 10 years ago. Here's another one, and that is mother's milk is not sterile. Even if you obtain the mother's milk from inside the mother's breast, it has bacteria in it. It was well known that infants, when they're nursing, they're sucking the nipple, and the nipple will have bacteria on it. And so it was well known that infants were getting bacteria from the mother from their nipple, but they're also getting bacteria from their mother's milk directly. And that was a surprise, that the milk inside the mother's breast has bacteria in it. Any question about any of that? Most of that bacteria is actually beneficial to the infant when they're getting the bacteria in the mother's milk. I'm not sure why this one was a surprise, because the placenta has a normal microbiota. And if you know anything about the female reproductive system, it is open to the outside. So it's not a surprise to me that the placenta could have microbes in it. It is a unique normal microbiota, though, where the placenta has a species and a configuration of species that is not seen elsewhere in the human normal microbiota. So it is a unique placenta microbiota that obviously only happens when the female is pregnant, but there is a normal microbiota in the placenta. Any question about any of that? All right. The normal microbiota is very important to the host, because the normal microbiota can engage in microbial antagonism with a pathogen. Antagonism is competition between two or more species, and the normal microbiota can compete with the human pathogen. Obviously, in the case of my skin, my normal microbiota competed with the Staphylococcus aureus and then threw off the Staphylococcus aureus from my skin. And another case where we have skin with the normal microbiota on it, if a pathogen were to fall on the skin, the space might be occupied by the normal microbiota, in which case the pathogen cannot get established. So you can occupy the niche the pathogen might occupy. You can compete for resources. For example, if the Staphylococcus aureus were to fall on my skin, and right next to that area would be Staphylococcus epidermidis, and the Staphylococcus epidermidis have used up all the resources in that region of the skin, that will not give any resources to the Staphylococcus aureus, so it would not grow and get established. The normal microbiota can also produce acid, making the region of the body too acidic for the human pathogen. This is especially true in the mouth and the urethra, but it's also true on the skin, where the normal microbiota make these areas more acidic. And that might be too acidic for a pathogen to get established, grow, and then cause disease. For example, the sebum is actually slightly acidic, and when the normal microbiota of our skin metabolizes the sebum, it makes it more acidic. And so our skin is actually around pH 5, and that is too acidic for many human pathogens. Any question about any of that? All right. Another way the normal microbiota can engage in microbial antagonism is if the normal microbiota produces a protein that we give it a name bacteriocin, which inhibits the growth of a pathogen. For example, E. coli produces bacteriocins that inhibit the growth of Salmonella and Shigella. Now, bacteriocins only inhibit the growth of microbes which are similar to the bacteria that made the E. coli. So an E. coli bacteriocin would inhibit the growth of Salmonella or Shigella, but it would not inhibit the growth of, oh gosh, what's the name of that? Something fecalis, Enterobacter bacteria fecalis, meaning a bacteria which is very prevalent in our gastrointestinal tract. So a bacteriocin would prevent the growth of Salmonella and Shigella, assuming it's coming from E. coli, but would not prevent the growth of many other species of bacteria. You can think of a bacteriocin as an antibiotic because it has a similar effect. The difference is it's much more specific than an antibiotic. The best we could do with an antibiotic would be to only stop the growth of gram negatives. And as you know, we have many gram negatives as part of our normal microbiota in our gut. So if you take an antibiotic, it will inhibit many of your gut normal microbiota cells. If, on the other hand, you were to take a bacteriocin, most of your normal microbiota in your gut would not be affected. And I've often wondered why we are not taking bacteriocins to treat with bacterial infections. And a few years ago, I read where people were experimenting using bacteriocins to treat bacterial infections. Any question about any of that? So bacteriocins are an experimental treatment now for treating for bacterial infections. All right, any question about microbial antagonism? If not, let's move on to probiotics. That is the consuming or taking of live microbes to exert a beneficial effect. For example, if you eat yogurt with live culture, meaning live bacteria in them, you're much less likely to come down with a yeast infection. Doesn't matter where the yeast is growing. If you eat live culture yogurt, you will be less likely to come down with a yeast infection. And it's because the bacteria and the yeast compete with the yeast. So you're much less likely to get the yeast infection. So probiotics is intended to exert a beneficial effect. And they're actually kind of common now. You can find probiotics in most grocery stores. Any question about any of that? All right. The normal microbiota establishes a relationship with the host, and because the two species are living together, we call it a symbiotic relationship. Symbiosis, all it means is two species living together. The normal microbiota forms a commensalistic or a mutualistic symbiotic relationship with the host. A commensalistic relationship is commensalistic symbiotic relationship is where one organism benefits by living together with the other, and the second is not harmed. Some normal microbiota do fit that category, where the normal microbiota are given a home, and they might even be given food by the host, and then the normal microbiota do not harm the host. They don't necessarily give it any benefit, but they do not harm the host. One example of a commensalistic organism, microbe, living with us is in each of our eyes, we have one microscopic nematode, which is a roundworm, and it lives in the eye and gets a home from us. And really, all we give it is a home. We don't feed it, but it probably consumes bacteria that fall into the eye because it does not eat human tissue. So it doesn't do us any harm. And anyways, that's a commensalistic relationship. There might be some normal microbiota living on our skin, which most of the time is commensalistic, where we give it a home, we might even give it food, so it benefits, but it doesn't do us any harm. It doesn't do us any good most of the time. But the thing is, every once in a while, it may compete with a pathogen, in which case it's doing us good. And that's not a commensalistic relationship. That's a mutualistic relationship where both organisms benefit. Like E. coli in our gut benefits us because it helps us digest our food and then shares the food with us. We, of course, benefit because it's helping us digest the food, and then it benefits because it's given a home and it's given food by us. And that's a mutualistic symbiotic relationship. And there's some normal microbiota that engage in a mutualistic relationship with us. Like I said, many of the skin normal microbiota, at least at times when they're competing with a human pathogen, that would be benefiting us. So there's some which I guess you could say, like Staphylococcus epidermidis, which most of the time is commensalistic, but sometimes it's mutualistic. And then you have a human pathogen. It forms a parasitic symbiotic relationship with us, where one organism benefits the pathogen because it's getting food, a home from us, it's damaging us, and it's doing harm to us. So that's a parasitic symbiotic relationship. And all human pathogens are parasites. They do us harm and they benefit by living on us. The normal microbiota does not normally cause disease, at least in healthy individuals. However, the normal microbiota can cause disease under certain circumstances, in which case we call it an opportunistic microorganism. A normal microbiota that becomes an opportunistic pathogen, which can cause disease under certain situations, such as if the patient is immunocompromised, they don't have the immune system to keep the normal microbiota in check, And then the normal microbiota, the species of the normal microbiota can cause disease, becoming an opportunistic pathogen. Other examples of opportunistic pathogens are E. coli, which is normally found in the gut. And when it's grown in the gut, it doesn't cause us problems. But when it's grown in the ear or in the lungs, it's not supposed to be growing there. And it will be an opportunistic pathogen if it grows in the lungs, causing us pneumonia. Staphylococcus aureus is found in about 50% of Americans in their nose and throat. And in the nose and throat, Staphylococcus aureus does not usually cause disease. But if it's growing elsewhere, like on the skin, it can cause disease, like a skin boil. And yeah, Staphylococcus aureus can be on the skin of 20% of Americans, but it's much higher on the nose and the throat of Americans, where about 50% of them have it. Fortunately, in the nose and the throat, it doesn't usually cause disease. All right, any question about any of that? If not, let's move on to Koch's postulate. We talk about this topic 3 times in the term, and I don't know if this is the second or the third time we've talked about Koch's postulate. But Koch's postulate was the steps Dr. Koch took to show that Bacillus anthracis causes the disease anthrax. To engage in Koch's postulate, you have to first obtain the microbe from a sick individual, or in this case, a sick bunny. And then you grow up the microbe in vitro and purify it. And then if you take that microbe that's been purified and inject it into a healthy host, in this case, a healthy bunny, and if that bunny could become sick, as this bunny did, and that's why the bunny is on its side, because it's a sick bunny. Oh, the poor bunny. Then that's indication that this microbe causes this disease. But Dr. Cox said you got to go one step further, and you have to show that out of this sick bunny, you can obtain the microbe that you injected into the bunny to make it sick. And if all of these steps are in place, this is Koch's postulates, the steps you use to show that a given microbe can cause a given disease. And if you use Koch's postulates for showing that a given microbe causes a given disease, you will not ever be wrong in finding the etiological agent, the agent causing the disease. Koch's postulate was not used when Louis Pasteur's lab came up with Haemophilus influenzae as the cause of the flu. What Louis Pasteur's lab did was he was sampling flu patients to see if any of them had a bacteria that might be causing the flu. And he found one that was, at least in some flu patients, and he named that bacteria Haemophilus influenzae. However, Haemophilus influenzae does not cause the flu, a virus does. And Louis Pasteur did not ever isolate the virus. He happened to get just a bacteria that happened to be present in a couple of patients of the flu. And so he did not use Koch's postulate, and he got the wrong organism. We still call the bacterium Haemophilus influenzae, even though it is misnamed, because it doesn't cause the flu. There are exceptions to Koch's postulates. For example, what do we have if the microbe has unique culture requirements and we cannot get it to grow in vitro for over 100 years? Let me think about that. Probably close to 140 years, they were trying to get the microbe that causes syphilis to grow in tissue culture. And they could never figure out what they needed to add to the media to get Treponema pallidum, the microbe that causes syphilis, to grow in tissue culture. And only recently, less than 10 years ago, have they finally solved this problem of what they need to add to the media to get Treponema pallidum to grow. So now we can get it to grow in tissue culture. But what did we do when we couldn't get it to grow in tissue culture? We knew that syphilis caused, Treponema pallidum caused syphilis, but we couldn't get it to grow in tissue culture. So how did we show that Treponema pallidum caused syphilis? Another microbe which we could not grow in tissue culture, and I haven't checked to see if we still cannot, but Bartonella henselae is the organism that causes cat scratch fever. And yet we never got it to grow in tissue culture. So how could we say that it was the organism that was causing the disease? Anyone have a guess? Think about that. What do you do if there's a disease which multiple organisms can cause the same disease signs and symptoms? For example, with the human cold, not only is there more than one species that can cause the human cold, but there's actually 3 families of viruses which can cause the human cold. So with these, you can't use Koch's postulate because there's more than one species that can cause the same disease signs and symptoms. And then what do you do if there's one species of bacteria or another species of microbe which causes more than one disease? For example, salmonella causes 2 diseases, salmonellosis and typhoid fever. Strictly speaking, you can't use Koch's postulate to show that salmonella causes either disease because not all organisms, not all cases will be caused by that organism. Although you could look at to see if all cases of salmonella are caused by salmonellosis, are caused by salmonella. And they probably have done that. And then all cases of typhoid fever are caused by salmonella. But the point is there's more than one disease that is caused by the same microbe. And so generally Koch's postulate is not used. And then what do you do if there are moral and ethical conditions for a human-only disease? Like AIDS, we think HIV causes the disease AIDS, and yet we've never injected HIV into patients to then later see if they develop AIDS. So how do we know that HIV causes AIDS if we've never injected HIV into a patient and then use Koch's postulate to show that HIV is causing AIDS? Anyone have a guess? What have we done? How do we know that HIV causes AIDS? Anyone? All right, you should think about that.
Have we injected it into animals to study it that way?
Not really, because HIV will only grow in one species of chimpanzee, and it doesn't cause AIDS in the chimp. So there'd be no way that we could inject it into the chimp and then later say that HIV causes AIDS in the chimp, because it doesn't cause AIDS in the chimpanzee. So that's a good guess. And they have done that for, I don't know, um, like syphilis. They can inject uh, Treponema pallidum into mice, not mice, into rabbits, and then get it, syphilis to occur in the rabbit. But it doesn't grow very well in the rabbit. And after a few generations, something like 3 or 4, it stops growing in the rabbit because it's not getting the requirements it needs from a human. It's getting some requirements from the rabbit, but not all the requirements it needs. Which, don't ask me what the requirement is, but it can cause syphilis in the rabbit, but only for a short time, few generations. And I assume that's generations of bacteria, but I don't know. It might be generations of rabbits. Anyways, how we do that is we do vast epidemiological studies, like we've studied large number of people, and then people who are HIV negative, we go back into the population and study them again. And some of those HIV negative patients became HIV positive. And then we go back at a later time and find that those HIV positive patients, some of them became AIDS patients, and only patients who are HIV positive were the ones who become AIDS patients. So that's how you can show that a given microbe causes a given disease without using Koch's postulate. And that is vast epidemiological studies, and you have to study the population over time. Any question about any of that? All right, if not, let's move on to some more terms. A symptom is a change in the body function that is experienced by a person as a result of a disease, but it is subjective. It is not apparent to an observer. An observer cannot measure the symptom. For example, when you have a headache, nobody else can measure or even know that you have a headache. Other than you're telling them, they have no way to measure that you have a headache. So a headache is a symptom that nobody else can know that you have that symptom except for you. So it is subjective. A sign, on the other hand, is a change in the body that can be measured or observed as a result of disease. So a sign is an objective measure. For example, if someone has a temperature of 38.5 degrees Celsius, and that's something over 100 degrees Fahrenheit, that temperature can be measured by somebody else. So that's why we call it a sign, not a symptom. And these are textbook definitions. You may or may not find that a clinician will use these definitions. And generally in the general public will use one for the other. But in reality, a symptom is one that you can experience, but nobody else can measure that you have it. And a sign is something that you can experience, but everybody else can measure that sign. Any question about any of that? A syndrome is a specific group or signs or symptoms that characterize a given disease, such as the signs and symptoms characteristic of AIDS. A communicable disease is any disease that can spread from one host to another. Examples: measles, tuberculosis, chickenpox are all communicable diseases. A contagious disease is a disease that can spread easily from one host to another. For example, a contagious disease is the flu or COVID-19, where the disease organism can spread easily from one host to another. Can anyone name a communicable disease which is not contagious? Meaning it can spread, but it doesn't spread easily. Anyone have a guess? It's actually one of these three right here. Does the measles spread easily? Is the measles contagious? Okay, how about chickenpox? Is it contagious? All right, you guys think about that.
Yes.
Yes, the measles is contagious. It does spread very easily. In fact, the measles is one of the most contagious diseases out there, even more contagious than COVID-19. Although COVID-19 is catching up, but it's not as contagious as the measles. The chickenpox is also a very contagious disease, easily spreading from child to child. In the old days, before the chickenpox vaccine, people would have chickenpox parties, and then people would bring their healthy children over and get them exposed to the chickenpox. And it was good because you want the child to get the chickenpox when they're healthy, as opposed to getting it when they're sick. And most of the human population got the chickenpox before the chickenpox vaccine came out. However, since the chickenpox vaccine has come out, the majority of children, at least in the first world countries, no longer get the chickenpox. That's because they're vaccinated. Tuberculosis is a communicable disease, but it is not contagious. Tuberculosis does not spread easily. An even better example is, oh gosh, now I dropped it. I just had it on the tip of my tongue too. Leprosy, there we go. Mycobacterium leprosy. The bacteria that causes leprosy, it does not easily spread from one host to another. In fact, almost 90% of humans cannot get that microbe to grow in them. Only about 10% of humans can. So it doesn't spread to most people, but it can spread. So it is communicable. A non-communicable disease is one that does not spread normally from one host to another. An example would be tetanus. It does not normally spread from one host to another, so we call it a non-communicable disease. Any question about any of that? Now, I said it does not normally spread from one host to another. There are some cases where you could think of a theoretical case where tetanus could spread. And that is if somebody were to get tetanus and then somebody else were to eat them, especially after not cooking them well, the cannibal could get tetanus. But other than that, tetanus does not spread from one host to another. So does not normally spread. Any question about any of that? And no, I'm not going to ask you a question, something like, can a non-communicable disease ever spread? And then list the case. I'm not going to do something like that. It's just a curious case of a non-communicable disease not being communicable. All right, when talking about the occurrence of disease, it's important to talk about the occurrence to get the full scope of the disease. You need details about its occurrence. The occurrence has two terms, the incidence and the prevalence, that describe it. Incidence is the fraction of a population that contracts a disease during a specific period of time. And the better way of saying it is the incidence is the new cases of disease only. The prevalence, on the other hand, is the fraction of a population having a specific disease at a given time. And the prevalence includes the new cases of disease and the old cases. So back in 2007, when we were talking about the AIDS in the United States, the incidence of new cases of AIDS in the United States in 2007 were 56,300 cases, came down with new cases of AIDS. But in 2007, the total prevalence of patients in the United States with AIDS was 1,185,000, because that includes both the new cases and the old cases. Any question about that? Some diseases, we tend to use one term over the other. Like in COVID-19, they usually talk about the total number of people in the United States getting COVID-19, in which case now I think it's way over 1 million. It may be up to 2 million now. And I think our population, I don't remember our population. I think it's around 6 million, but maybe it's more. So a large number have gotten it. Which actually I wouldn't be surprised if over 50% of the population have gotten COVID-19 now. So it's probably over 3 million. So some diseases use prevalence. Other diseases, like usually when they're talking about the flu, they're giving incidence. The number of flu cases so far this year, they usually talk about incidence. Any question about any of that? Some other terms when we're talking about the occurrence of disease, a sporadic disease is a disease that occurs only sporadically in a population. Like in the United States, typhoid fever is said, at least by some people, to be a sporadic disease. It occurs only occasionally. An endemic disease is a disease that is constantly present in a population. For example, the human cold is an endemic disease wherever you are in the world, because common cold is frequent everywhere, especially in the wintertime. However, we could say malaria is endemic to the tropics, because malaria normally only spreads in the tropics. And that's where we find the disease. An epidemic is a disease acquired by many hosts in a given period of time, a short period of time. An epidemic is usually also only on one continent. So when COVID-19 first started in China, it was an epidemic. And as long as the disease was limited to China, it was an epidemic to Asia. However, COVID-19 soon spread to the rest of the world, in which case it became pandemic, or worldwide spread of the disease. An epidemic is a disease where the incidence is higher than expected. Like normally in the wintertime, we have higher cases of the flu. That's not necessarily an epidemic or a pandemic, because you have to have significantly higher numbers than expected to be an epidemic or a pandemic. And we get a epidemic of the flu about once every 10 years, where the numbers are greater than normal. And what happens is the flu virus changes about once every 10 years, and the new flu is one that hasn't been seen before, And that's why it becomes an epidemic, because so many people are susceptible to it. And there is no immunity because it's a new variant of the flu. Any questions about any of these terms? Now, when we're talking about low numbers of disease, it's arguable whether the disease is sporadic or endemic. Like some people would say, no, we always find typhoid fever in the United States at low numbers because we always find it's an endemic. And other people will say, no, it's sporadic. And when we're talking about low numbers, you'll get into the argument. We're not going to talk about that argument, and we're not going to discuss what typhoid fever is. Maybe a better example of a sporadic disease is every once in a while in the United States, we have malaria in the United States. And last year, it actually was endemic in Texas, Florida, and one other southern state where the malaria was spreading by the mosquitoes in the South, meaning we have the mosquito, at least in the Gulf states in the South, that can spread malaria. And somebody got malaria from a from the tropics and brought it back to the United States. A mosquito bit them, and then the mosquitoes were spreading malaria. And hopefully this next summer, malaria will be gone in the United States, so it won't spread again. But there were like 6 cases in Texas and over a dozen cases in Florida where people were getting malaria from being bitten by mosquitoes. Normally in the United States, we don't have malaria spreading. And that's just because the cases of people having malaria in the United States are very rare, but we do get it sporadically when somebody leaves the United States, picks it up from the tropics, and then brings it back to the United States. And then it can spread in the South, but usually it's not present, so it doesn't spread. Any question about any of that? Another useful way of defining the scope of a disease is to talk about the terms of the severity or duration of the disease. An acute disease is a disease where the symptoms develop rapidly. The disease is short-lived. It runs its course and then goes away. An example would be the human cold. The symptoms develop rapidly. The disease is short-lived, and then the disease goes away. A chronic disease is a disease that develops slowly. It persists for long periods of time. It usually gets worse with time. Mononucleosis is a chronic disease. Hepatitis B disease, where the liver will become inflamed, and then the patient gets hepatitis B inflammation again and again. A chronic disease. Some chronic diseases can go away. Mononucleosis eventually goes away, but it takes a long time, several months. Hepatitis B disease, you could get that lifelong, where the chronic disease just gets worse with time. More and more damage to your liver will occur. There are diseases in between that. We're not going to discuss it. It's called the subacute disease. You don't need to know that. All right. Any question about any of that? If not, I'm going to end here. I will be in the lab, but only to answer questions. And I'll only be there for 15 minutes. Any questions before we go? All right. I'll see you later. Bye.